Welcome everyone. My name is Jeremy Gagnon. I just started my master in Sébastien Rodrigue Laboratory, and today I'm going to present my project about the construction of a synthetic Mesoplasma florum genome. Since Mesoplasma florum has already been introduced by my colleagues, I will simply remind you that M. florum is a molecule located in the phylogenetic tree between Mycoplasma and Spiroplasma groups. The project that I recently started has two main objectives. I will first develop a protocol to rapidly build Mesoplasma florum synthetic genomes as an artificial chromosome in the yeast Saccharomyces cerevisiae. And I am also interested in determining how many DNA fragments can be transformed and assembled at a time in yeast. After the initial demonstration of that we can build a synthetic genome, we will start generating variants, for example, by reducing the genome section by section. We will then use genome transplantation to boot up and bring them to life, as previously done by the GCVI team. Our strategy is to divide the Mesoplasma florum genome into 16 sections, each of approximately 50 kb. And after that, each section will be assembled using 12 fragments of about 4.1 kb. And in total, the genome consists of 189 fragments. The fragments for this first synthetic genome are all amplified by PCR using primers designed with the Primer3 software and our in-house set of bioinformatic tools. And all of the primers must be devoid of hairpin, homodimer, heterodimer, and non-specific binding. The primers also have a GC clamp at their 3' end and a melting temperature between 53 and 60 degrees Celsius. The fragments are designed to have an average of 80 base pair in homology with the adjacent fragment in the genome. And here are an example of how the primers are designed to allow overlapping with each other. So we can see the fragment 103 that's going to the right and the fragment 102 that's going to the left to bring an overlapping with the two of them. After the first attempt to amplify the 189 PCR reaction fragment, we have a success rate of 94% with only 11 negative reactions. The negative reactions are all successful after additional attempts. And after all the amplification, the PCR are purified using spray magnetic beads, and their concentration are measured to ensure that we have at least 100 nanogram of DNA per fragment for the following steps. To assemble the genome sectors in yeast, we need a vector to select the products. This vector is split into two fragments to reduce the chance of recircularization without the Mesoplasma florum genome section. The first vector fragments contain ARS for autonomously replicating sequence, a centromer, and the promoter of the IS3 gene. The second part contains IS3 coding region, and another auxotrophy marker, URA3, is located at the opposite of the construct. And each side of the vector and URA3 has 80 base pairs of homology with the adjacent fragment. We use Saccharomyces cerevisiae VL648 for the transformation and assembly of the genome sectors, and we use two protocols for the transformation. One using lithium acetate and polyethylene glycol, and the second using spheroplasts that are created by removing the yeast cell wall with zymolase. And for now, the PEG and lithium acetate appears more efficient, but we know that the procedure using the spheroplast can be further optimized and will likely more efficient for larger construct, and that can be that may be helpful for the next part of the project. So after the transformation, the resulting construct needs to be extracted from the yeast and validated by sequencing. After the validation, the URA3 marker is removed by homologous recombination with a G block. And we next use the restriction enzyme NOT1 to cut at both sides of the vector and release the assembled genome sections. And after the first set of transformation, 16 assembly products will be generated, which opens different possibilities for the next step. We can either individually test the sectors in the otherwise wild-type genome, or we can transform yeast with 16 fragments at once to reassemble the complete genome. And after we have obtained and confirmed the florum synthetic genome sequence, we will transplant it into a recipient M. capricolum cells, as previously reported by our group and the group of Carol Lartigue. We also plan to use a florum W17 strain that we know that is phylogenetically closer than M. capricolum, and that has a great transformation rate compared to M. florum L1. So this is how we plan to proceed to build M. florum synthetic genomes. And for the next steps, we can think about modifying a section of the genome and see what consequence this produce and try to move towards the reduction to a minimal genome. So with that, I would like to thank my group for their help and the support with my project. And thanks for your listening. And I'm ready to try to answer any question.
You guys are ambitious. I can't wait to see. You know, in theory, I mean, it worked for us. Why not you?
Yeah, we will see.
And remember, we got 25 pieces to go together to make Mycoplasma genitalium, and we did some calculations, and by Hamsmith's calculations, he thought it would max out at about 52 pieces to put together with yeast. So I think, you know, I won't say it's a sure thing, but I expect next year to hear about your fully synthetic Mesoplasma.
We expect to. We will see.
Other questions? Anyone? When do you think you'll have an answer?
Maybe the end of winter, start of the winter. Mm-hmm. We will see how it's going. We don't know.
I urge you to think about sequencing your pieces. We had a single base pair, a single base pair missing in DNA A, and we couldn't figure out why the genome wasn't working until we went back and resequenced and found that, you know, we were dead before we ever started. Okay. Jeremy, thank you very much. Everybody, thank you very much.